Assalamualaikum and good morning to the judges and audience. I'm Sabrina Faizal. Along with my partner Sabrina Asri, we will present a poster entitled Systematic Review of Experimental Studies Evaluating the Association of Sugar and Sugar Substitute on Solid Tumours. Cancer ranks as the top leading cause of death globally. This poses a significant impact not just on the individual but on the government due to significant economic loss. On top of that, the statistical world cancer trend is escalating and expected to increase due to unhealthy lifestyle, which is one of the contributing factors of cancer. However, the association between sugar and cancer remains unknown. In general, there are two types of sugar available in the market. The most common consumption is dietary sugar, a nutritive sweetener that supplies adenosine triphosphate, which is ATP. And the least common is sugar substitute, a new generation of sugar invention created to cater for the demand of certain diseases such as diabetes mellitus and obesity. Also, it's a non-nutritive sweetener, thus provide a non-ATP supplies on cells. However, is it safe to consume? The fact that cells requires energy to maintain cell integrity is widely known. This energy is generated within mitochondria through a biochemical process which at the end produces ATP. The nature of cancerous cells is that it proliferates fast, thus requires more ATP. Therefore, researchers are keen to know whether cancer cells will bypass mitochondria to fuel their demand and survival growth, or vice versa. Thus, the hypothesis of sugar fuels cancer cells was generated. Sugar fuels cancer. Is it myths or facts? Is the impact of sugar and sugar substitute on cancer proliferation similar or different? Evidence of association between sugar and cancer risk in preclinical studies has been acknowledged as a reliable tool to provide a clear scientific answer with strong reasoning and justification. Thus, a systematic review of preclinical studies assessing the sugar-induced DNA damage, cancer cell proliferation, viability, apoptosis, and angiogenesis, which are the hallmarks of cancer highlighted by Hasnah and Winberg in 2011, might be a good measure revealing the effects of sugar and cancer. In this systematic review, 3 solid tumors were highlighted. Breast and lung cancer was chosen because it was ranked as the 2 top cancers according to Global Can 2020. Oral cancer was ranked as the 18th most common cancer worldwide, but it is included in this review because the cases are usually underreported and notably the most common cancer that affects the patient's quality of life tremendously. Objective of our review is to be aimed to systematically review preclinical studies that evaluate the association of sugar and sugar substitutes on breast, lung, and oral cancer development and progression, with intervention aimed to reduce cancer risk, mortality, and morbidity. Now, the method to perform these systematic reviews is as depicted in this flow diagram. First of all, formulated research question was designed using the PICO method. Firstly, the population include breast, lung, and oral cancer in vitro or in vivo study. Next, the intervention is treatment of supplementation of dietary sugars or sugar substitutes on tested cancer cell lines or biological models. Comparison is done between the laboratory results of several experimental studies after sugar and sugar substitute supplementation. Laboratory parameters assessment which was compared includes cancer cell proliferation and DNA damage. As for the outcome, the primary outcome of this review is initiation of cancer upon sugar supplementation, and the secondary outcome is sugar-induced cancer cell progression. The Boolean search term was established and 3 electronic databases which are Scopus, PubMed, and EBSCO were used. Inclusion and exclusion criteria screening were conducted. To assess the methodological quality, all steps were performed in duplicate independently by the first 2 authors and re-evaluated by another 2 authors following the standard Cochrane Handbook for systematic reviews of intervention. This prisma flow diagram method is used to portray the flow of the review. First, in the identification phase, retrieved articles were assessed and following these criteria. First, the article was published from August 2010 up to August 2020. The study is worldwide. Participants included are preclinical studies, either in vitro or in vivo. The parameters assessed are DNA damage and repair, cell proliferation, and cell viability of cancer cells. Publication type is an original article. With that, total records identified is 221, with 14 papers from Scopus, 95 papers from PubMed, and 112 papers from EBSCO. In screening phase, intra-duplication and inter-duplication title screening were conducted within and between databases. No papers were excluded. After that, following the exclusion based on title, 64 papers were excluded and the remaining papers were 157. In eligibility phase, following exclusion based on abstract, 143 papers were excluded, only 40 papers remains to be assessed. Lastly, in included phase, only 14 papers met all those phases in PRISMA guidelines and were included and reviewed. Analysis of each article was carried out, compared and assessed.
A total of 14 preclinical studies were identified and fulfilled the inclusion criteria. Major preclinical studies were focused on breast and lung cancer. Only one experimental study reported on oral cancer. 6 studies reported glucose and or sucrose-induced cancer progression, while 7 studies reported that sugar substitute inhibit cancer proliferation, subsequently acting as potential cancer therapy. Another study on sucrose and xylitol reported the association of cancer promotion and inhibition respectively. The synergistic effect of DLOs or cisplatin on cancer was also mentioned in a study. The summary of the article's analysis was summarized in our paper. This systematic review, there are 2 main findings. First, the association of sugar on cancer progression and proliferation. Most sugar substitutes, either natural or synthetic, demonstrated anti-tumor effects against cancer progression and proliferation. Aspartame was the only sugar substitute that portrayed a different result. The aspartame supplementation facilitates pathological conditions characterized by inflammation. Thus, it is a potential angiogenic agent that can induce regenerative cytokine production. The supplementation of the rest of the sugar substitute significantly inhibits tumor cell proliferation and promotes apoptosis of selected cancer cells. To evaluate the anti-tumor properties, apoptosis and cell proliferation assays were utilized. The laboratory output results show good evidence either quantitatively or qualitatively. These act as a reliable experimental tool to observe apoptotic cell development as well as evaluation of cell proliferation suppression. Finding of an indirect association of sugar metabolism and the increase of developing cancer were also evidenced in some studies. A study conducted by Iconlab et al. In 2018 showed that dietary sugars promote lactate dehydrogenase (LDH), which indirectly induce neoplasia. Other findings are via the ability to induce cell transformation, facilitate pathological conditions characterized by inflammation, potentiate angiogenesis and oglynisacylation of G6PD that have been reported by Aleva et al. In 2011 and Rao et al. In 2015. Second, the association of sugar and genomic DNA. According to Iskel et al. 2020, sugar substitute induce single-strand DNA damage on a cancerous cell in a dose-dependent manner. Hence, the amount of DNA damage is inversely proportional to cancer survival. The unexpected finding of this systematic review was sugar substitutes such as DLOs in a combination of cisplatin promotes synergistic effects. Therefore, the cisplatin resistant issues might be tackled up via this discovery. The limitation of this systematic review are the selected papers were mainly focused on analyzing the experimental methods, assessing sugar and sugar substitute-induced cancer cells. Apart from that, only 3 solid tumors were chosen, which are breast, lung, and oral cancer. Only 3 databases were selected due to the restricted access to the online databases provided by the institution. In conclusion, our systematic review reveals that sugar potentially possesses anti-tumor properties and also stimulates cell proliferation. These findings highlight dietary sugar potentially proliferates cancer cells due to production of energy. Sugar substitutes are a non-ATP source, thus possessing a cancer protection effect. In addition, the output results depend on the cancer type. Long-term expectation of our systematic review is creating a new potential oncology research area. This will eventually benefit in improving cancer treatment as well as supportive care for cancer patients.
With that, thank you.